Hello and welcome back, everyone. I hope you all are doing good. So, today we'll be learning about a very new topic in the epigenetics series, and that is the methods to detect DNA methylation. So, till now, we have learned about DNA methylation, DNA demethylation. So, how to know that in the genome whether the DNA is methylated or not? So, we can carry out certain sequencing procedures which will help us to know whether DNA is methylated or not. So, today we'll be learning about bisulfite sequencing. So this bisulfite sequencing is a procedure, is a sequencing method in which we get to know whether the genomic DNA is methylated or not. So till now we have learned that the fifth carbon of cytosine on this fifth carbon, if we add CH3 group, it gets converted to 5-methyl cytosine. And the enzyme which is going to carry out this reaction is DNMT, DNA methyl transferase. So there are different members of this family, methyl transferases family. So the DNMT1 is maintenance methyl transferase, while DNMT3A and DNMT3B, those are de novo methyl transferases. So we have learned all of this in the DNA methylation video lectures. You can refer to that. So, how to know whether the genomic DNA is methylated? So, in the normal sequencing methods, you will only get the idea of the what is the sequence of the DNA. Like, for example, you carry out the sequencing on this DNA strand. Okay, this is a DNA strand. Okay, this is single strand of DNA, and you carry out the sequencing. Now you will know what's the sequence A A T G C G C C T A. Okay, but in the normal sequencing procedures, you want to know whether this cytosine is methylated or not. So for knowing that whether the CH3 group has been added on this cytosine or not, we do bisulfite sequencing. Okay, so let's learn about this sequencing method step by step. First, we in the for this first we denature the DNA. Okay, so DNA denaturation happens at a very high temperature at 95 degrees Celsius. So due to that, you'll get nice single stranded DNA. So now after the DNA denaturation at low pH, okay, so about 5 pH, you add sodium bisulfite. Sodium bisulfite. Okay, so sodium bisulfite is NaHSO3. This is sodium bisulfite's chemical formula, and you add sodium bisulfite. So now this sodium bisulfite will do the conversion reaction. Okay, so now what conversion reaction? It will deaminate the cytosine. So now what does deamination of cytosine results? It results into uracil. Okay, deamination of cytosine will result into uracil. So, due to addition of sodium bisulfite, the cytosine on the DNA strand gets converted to uracil. But if instead of cytosine, it is 5-methyl cytosine, means the CH3 group is added on it. So now this is not able to undergo deamination. Deamination reaction won't happen because sodium bisulfite is unable to deaminate 5-methyl cytosine, and it will remain 5-methyl cytosine. Okay, so in the normal, we learned that in the normal sequencing reactions, we won't know whether it is cytosine or it is 5-methyl cytosine, right? We won't know that. But now here in the bisulfite sequencing, you have converted the cytosine to uracil, and this uracil is detected as thymine in the sequencing reactions. Why? Because uracil is component of RNA, right? So it is read as thymine in on that DNA strand during the sequencing reaction. May it be Sanger sequencing or some other Maxim Gilbert sequencing, whatever sequencing reaction you are using. So this uracil is read as thymine. So first step of this is denaturation. The second step is the conversion. This is the second step. And the third step of this reaction is desulfonation. So now what you do, you just remove the sulfate moiety which is already present. So it is the removal of sulfate moiety. Okay, so how do you do desulfonation? It is also known as alkylation. Means addition of base. So addition of base results in removal of sulfate moiety, which was added on the which results in deamination of cytosine to uracil. So the third step of the reaction is desulfonation. First we do denaturation of DNA, then at low pH we add sodium bisulfite, which results in conversion of cytosine to uracil, while it have no effect on 5-methyl cytosine. Okay, and the third step of this reaction is desulfonation. This desulfonation is done at high pH. Okay, so high pH means basic pH. So that is why it's also known as alkylation. So now you have removed the sulfate moiety, and now this genomic DNA strand, this is ready for sequencing. So now what you will do for the purpose of sequencing, this DNA strand needs to be amplified. You can't carry out the sequencing reaction from single DNA strand, right? The single genomic DNA. You need to first amplify it. So for amplification, you carry out PCR and then you go for sequencing. So now let's assume that you have extracted genomic DNA from some animal. Okay, so now the from human, let's say from human you have extracted the genomic DNA, and we are diploid organisms, so we have two sets of genes, so we have two alleles, right? So let's say this is allele 1 and this is allele 2. So let's say allele 1 is methylated in the genomic DNA, while allele 2 is unmethylated. Imprinted genes are methylated. So we'll learn about imprinted genes in some other video. Let just know that in the human genome, certain genes are methylated only on one allele. Okay, so like if only the paternal allele is methylated, or only maternal allele is methylated. So those are what we call as imprinted genes. So just let's drop that details for now. Let's assume that allele 1 genes are methylated while allele 2 sequence is unmethylated. Okay, so this is let's assume that the sequence is AAECG. Okay, and CCETA. Okay, so this is the sequence. Okay, let's just paste it here. So this cytosine here, this is 5-methyl cytosine. Okay, why? Because this is methylated while this is not methylated. So I'm writing simply C here. So the allele 1 is methylated while allele 2 is not methylated. So now what you'll do the bisulfite reaction as we learned above. So you will add, so now this is denatured. Okay, this is denatured genomic DNA, and you will add the sodium bisulfite. Okay, and so now it will convert the cytosine, to cytosine to uracil. So okay, oh, just a second. Mm, give me a second. Okay, so this 5-methyl cytosine will still remain as 5-methyl cytosine, because we know that this sodium bisulfite is unable to deaminate 5-methyl cytosine, while this cytosine, which was not methylated, it will be converted to uracil. Okay, so now you do the PCR. So for PCR, for the PCR of methylated sequence, you will have to take, different types of primers. So now see, for example, the primer is a oligonucleotide, right? So it could be 8 to 10 base pair long, or it could be 20 base pair long. For example, if it is 20 base pair long, so it also covers some C, right? Uh, what I'm trying to convey is that, so this sequence is not, so, it's a long sequence. Okay, it's a long sequence, and you want to amplify a certain gene. So in all those gene, you have to amplify this gene. Okay, and this is the whole sequence of DNA. Okay, so this is the whole sequence of DNA, and you want to amplify only this region. Okay, you want to amplify only this region. For amplification of this region, you will need primers, the forward primer and the reverse primer, right? So again, those primers, in those sequences of primers, there could be CG dinucleotides, right? And those CPG dinucleotides can be either methylated or not methylated. And that is why, while doing the PCR reaction, you will add different sets of primers. One set will be, so you will add methylation specific primers. Okay, so those in those primers, you will simply have the complementary sequence of C will be G. Okay, while in the unmethylated primers, the primers for the sequences which are unmethylated, you will in instead, see what I was trying to say. If assume that this is the primer here, and you're designing a primer for this sequence until C. Okay, so you're designing a forward primer for this sequence. So while designing it, it will be complementary, right? So you will design it like T T A, and instead of C, you will have G C G G. Okay, so this is methylation specific primer, because in the methylation specific primer, this C is not getting this 5-methyl cytosine is not getting converted, it is not getting deaminated. But in the unmethylated sequences, the C is getting converted to uracil, right? The cytosine is getting converted to uracil. That is why, while designing a primer, you will have T T A, and this U is read as T. So you will have A, C, G, G. This will be the primer sequence. So you will treat, instead of base pairing, instead of treating the C as cytosine as cytosine, you will treat cytosine as thymine. Okay, and that is why you are putting A in the primer. Are you getting it? Why? Because this cytosine is no longer a cytosine, it is converted into uracil. And in the uracil gets base paired with the adenine. Okay, that is why those primers, you will put the two sets of reverse and forward primers. One will be methylation specific primers, and other will be non-methylation, or non-methylation specific primers. Primers specific for unmethylated DNA. So you will have 4 different primers, right? A reverse primer and forward primer. Just a second. A reverse primer and forward primer for methylation specific, and a reverse primer and a forward primer which is unmethylation specific, right? So you will have 4 different primers, and you will carry out the PCR. Now after carrying out PCR, you will do normal sequencing steps, the normal Sanger sequencing steps. Okay, and now you will see that this will be read as, this in the sequencing will be read as AAT, and this 5-methyl cytosine will be read as cytosine. Obviously, in the normal sequencing procedure, we cannot differentiate 5-cytosine and 5-methyl cytosine, and that is why we are turning the cytosine to uracil, so that we can differentiate between the normal cytosine and 5-methyl cytosine, right? So this 5-methyl cytosine would not be read by normal sequencing method, and it will be read as cytosine only. Okay, so they won't be able to say that okay on this cytosine whether it has CH3 group or not. So it will be like AATC and for the sequence CGCCTTA. Okay, and while this sequence will be read as, so now it will be read as AAT, and the uracil in the normal sequencing methods is read as T, because this is DNA sequencing, right? This is not RNA sequencing. So in the DNA there is no uracil, so this will be read as T G C C T T A. Now you get two results from the sequencing. So you will know that whenever the cytosine is read as cytosine, okay, so this means that this cytosine was methylated. But when this cytosine is read as thymine, you will get to know that this cytosine was not methylated. Now let me explain it again. See, in so the cytosine gets converted into uracil, okay, due to bisulfite sequencing. Okay, while 5-methyl cytosine remains as 5-methyl cytosine. Okay, so now the sequencing methods, what happens that after that we do sequencing. Okay, so now we are doing sequencing. Now in the sequencing, we can't differentiate 5-methyl cytosine and cytosine. Okay, so whether it is 5-methyl cytosine, it will be read as cytosine. Whether it is cytosine, of course, it will be read as cytosine. So cytosine will will be identified as cytosine, right, in the sequencing. But 5-methyl cytosine will also be read as cytosine. Okay, so now this due to the bisulfite sequencing, this cytosine is converted into uracil, while 5-methyl cytosine is as it is. Okay, so now this uracil is read as thymine in the sequencing, now in the normal sequencing, but this 5-methyl cytosine is read as cytosine. Okay, now you get it. So whenever in the sequence, so now let me tell you this is the, so let me give you a question. So this is A T G G C G G G T T C G C C A T T. Okay, just a second. I'm sorry. Back to the topic. So this is the sequence. Let's assume that this is the sequence, and now this. So we, how many CpG dinucleotides are there? So we have this CpG dinucleotide, and we have this CpG dinucleotide. Okay. So we have two CpG dinucleotides, and let's assume that is this is methylated. This is 5-methyl cytosine, but this is not methylated. Let's assume that this is methylated and this is not methylated. Okay, but we don't know. So we let's see the differences when you do normal sequencing and when you do bisulfite sequencing. So what will happen in the normal sequencing? You will get to know that this sequence will be same. So in the normal sequencing, you won't be able to differentiate between 5-methyl cytosine and the normal cytosine, right? So what will be the result? In normal sequencing, you will get to know that the DNA sequence is something like this: A, T, G, G, C, G, 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 T, T, C, G, C, C, T, T. Okay, so this will be the result. So now in the normal sequencing, you don't know which C is methylated and which C is not. So you somewhat this seems like both are unmethylated. Okay, so there's no differentiation. There is no difference in the results. So you won't be able to decipher which cytosine is methylated and which cytosine not. That's why we abandoned this method and we moved to bisulfite sequencing. So in the bisulfite sequencing, what you will get? So this 5-methyl cytosine is not deaminated. So it will remain as it is. So it will be A, T, G, G, and this is 5-methyl cytosine. Okay, and due to bisulfite reaction, this will be G, 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 T, T, and this C is not methylated, and that is why it is converted to uracil. Okay, and this will be G, C, C, T, T. Okay, so now this is the bisulfite reaction. Okay, you do the bisulfite reaction, and then now you are doing bisulfite sequencing. Okay, you are running out the sequencing procedure. So during the sequencing procedure, you got to know that this is A, T, G, G, and now this cytosine is 5 5-methyl cytosine, but you are carrying out the sequencing. Okay, so it will be read as cytosine. So this cytosine will be read as cytosine, and then G, 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 T, T, and this uracil will be read as thymine, and then normally G, C, C, T, T. So now you got to know that this C is methylated. Why? Because it got protected. It didn't change. And this C, this extra T that we are getting that we are getting. So now when you compare this result, okay, when you compare this result with this result, you got to know that see, you got to know that here the C, here normal sequencing shows C, and even the bisulfite sequencing is showing C. So this means that this cytosine is methylated, while here this cytosine is normal. In the normal sequencing it is showing cytosine, but in the bisulfite reaction, bisulfite sequencing, it is showing thymine, means this is unmethylated. Okay, so the results will be, so whenever a cytosine is read, so a cytosine, so now let me give you a table which will help you out. So you in the normal sequencing, what you will see, and in the bisulfite sequencing, what will you see? Okay, so in the normal sequencing, the cytosine will be read as cytosine only. Okay, and this cytosine which will be read as uh, thymine in bisulfite sequencing. And in normal sequencing, 5-methyl cytosine is also read as cytosine, but 5-methyl cytosine is read as cytosine in the bisulfite sequencing. So now you here you get to differentiate. So whenever in the sequence, whenever for a DNA strand, you carry out both of the sequencing reaction, normal and bisulfite sequencing. By comparing the results, you got to know whether the cytosine is methylated or not methylated. Just like here, you got to know. Okay. So just like here in the normal DNA sequence, the C, and in the normal DNA sequence it was C, and in the bisulfite it was T, means this is unmethylated cytosine. Okay, and in the normal sequencing, if it is C, and again even if after doing a bisulfite sequence it remains C, means that cytosine was methylated. I hope this was clear, and in next video lecture we'll learn about oxidative bisulfite sequencing. Till then, see you. Bye bye.